This is the third part of video presentation in the transcription series. Here we will discuss the transcription machinery used in eukaryotic system. The machinery here refers to the various components and sequences associated with the transcription process. In the previous videos, we have discussed about bacterial mechanism in detail. So here we will see what are the different proteins associated and also the unique sequences which are significant in gene expression or in particular RNA synthesis in terms of eukaryotic system. The general transcription machinery in eukaryotes consists of various factors, of which we know the RNA polymerase enzyme is the vital molecule, of course, for transcription, and apart from that we have a number of proteins acting on the gene expression, which are generally called as transcription factors. And additionally we have the promoter regions, which is somewhat similar to that of bacterial system, except that this is little more elaborated in terms of their consensus sequences and number of positions. And at this, the third one or the finally we have the regulatory sequences, which are somewhat unique in eukaryotic regulatory mechanisms. Okay, so these are the four important factors we will discuss in this part of presentation. We know that RNA polymerase is the enzyme responsible for the synthesis of RNA or for the transcription reaction. And there are thousands of genes in eukaryotes, unlike bacterial systems. We know that when we take an example of human cell, we have approximately 20 to 25,000 different genes are there. Even though these 25,000 genes are representing only 1.5 to 2%, that's another story. But still the number of genes are pretty high in a eukaryotic system, and all those genes has to be transcribed as and when needed. So all these genes will be utilizing the limited transcription machinery for a transcription event. So which gene to be transcribed at what time and at what rate is the important regulatory event in the transcription process. So there are specific DNA binding proteins called transcription factors, which will mediate actually which gene has to be transcribed and thus a transcription event. So what actually these transcription factors do? They'll simply interpret the informations present in the gene promoter sequences and also in the additional regulatory sequences within the chromosome to the transcription machinery, in fact to the RNA polymerase enzyme, so that a successful transcription can onset or initiate. So these regulatory sequences are quite important in a eukaryotic system because the eukaryotic chromosome is a highly organized condensed structure. So when a gene has to be transcribed, well before the RNA polymerase enzyme is going to bind onto the particular gene location, that location has to be unwound properly so that only the RNA polymerase and other transcription factors can identify that sequence and go and bind over there. So these unwinding event, or otherwise we can say there is a decondensation process, and these decondensation events are regulated usually by these regulatory elements and associated proteins. So this is somewhat unique in eukaryotic system, and all the eukaryotic systems like yeast, from the single cellular yeast to high level eukaryotes, you can see the regulatory elements at various intensities. In yeast you can see one or two such regulatory sequences pretty close to the promoter region, that means to the core promoter region. But in higher level plants and animals, you can see there are diverse such sequences which are scattered up to a distance of around 10 kb. You have to remember that the length of a gene is not this much. The length of a gene is only maybe 2 to 3 kb only. But you can imagine the importance of regulatory sequences, because they are usually scattered up to around 10 kb, which means that the regulation is quite important. Because for a 2 kb or 3 kb gene, there are regulatory sequences which is spanning a distance of around 10 kb. So you can imagine the importance of such sequences. This image will give you an idea about such sequences. You can see in the upper part of the image, you can see which is of the yeast chromosome. There is only a single promoter region called core promoter, which is actually the Tatabox, and only one regulatory sequence, or one or two. You can see there is an upstream activator sequence or UA sequence and a silencer region. So there is the limited number of regulatory sequences are present in the yeast system. Whereas in the lower part of the image, you can see a much elaborated kind of these regulatory sequences. So apart from these core regions, which is up to the Tatabox, you can see the proximal promoter element, the silencers, enhancers at various locations, like even in the upstream and downstream you can see enhancer sequences, and additionally you can see insulators, and of which few of them are having a positive impact on transcription, and others will have a negative impact on transcription. So these all together, all these sequences are controlling the gene transcription or the RNA synthesis. Let's see how all these sequences and the proteins are interacting each other. You can see, suppose this is the DNA sequence or the part of a gene covering the promoter region. Suppose this is the core promoter region, maybe, and usually we know this part of the chromosome, that means the core promoter region of a gene, is the site for recognition by the transcription factors, which we have seen in similar to that in bacteria. The transcription factors will occupy this particular site. Usually there are different transcription factors that we will see soon, and these different transcription factor also will recruit the RNA polymerase enzyme, which is in blue color. So these RNA polymerase enzyme, once occupied, the system is ready for the transcription event. But in eukaryote, this is not over, or this is not enough for a successful transcription, or for the a kind of a high rate transcription mechanism. This is not enough. So additionally we have certain sequences also, so that we will see. That's what we have noted, like the regulatory sequences. So here I am pointing, or I am drawing two such regulatory sequences. Maybe it may be an enhancer sequence, or it may be an activator sequence, whatever it is. These are regulatory sequences. So these regulatory sequences are also occupied by certain proteins involved in the regulation event. They are what we call the regulatory proteins, or you can otherwise name them like activator or enhancers, etc. We have different sets of such regulatory protein. So these proteins will bind over these kind of sequences. But you can note that the binding is currently not affecting, or there is no interaction between the first set of proteins and polymerase enzyme to these proteins, which is on the regulatory sequences. Now still this particular gap is filled, or the interaction is made possible with the help of some other proteins. That's what we call the mediator sequences, or the mediator protein. So these mediators will play an important role for the connecting all those elements together, and once this is happened, this causes a successful initiation of transcription in a eukaryotic system. Now we are moving into a brief discussion on the basic features of all the listed factors. So we'll begin with the core promoter region. So the core promoter is nothing but a particular length of sequences which will be overlapping the +1 start site of a gene, and usually there are certain consensus sequences. That means certain sequences which are commonly found in all different organisms and for different set of genes. And such a sequence is what we call a core promoter. Usually the length of this sequence is usually around 60 to 80 base pairs in different organisms. And this is the site, the importance is that this is the site where the RNA polymerase and also the general transcription factor will come and occupy for a successful initiation. So there are sequences in the upstream site and also in the downstream site. That means you can see certain sequences in the upstream. That means they include the Tatabox, the BRE sequence, and also the initiator sequences. There are downstream sequences like MTE element or the motif 10 element, then also the DPE, that means the downstream promoter element. And you can also note down the position which is seen in the chromosome in comparison with the +1 site, and also given the transcription factors concerned which are interacting with that particular site, and also the specific sequence or the consensus sequences which are commonly found in that region. Yes. So we will start the discussion with the first element called the INR or the initiator element. So the INR is the initiator element. As the name indicates, this covers the +1 site, and it is somewhat similar to the Tatabox, which is the major promoter sequence in eukaryote. And the scientists have reported that this is the most common promoter which is seen when they analyze different types of genes. This is the most common promoter seen along with a gene, and usually these are recognized by two subunits of TF2D. And TF2D is important because TF2D is the first element or the first transcription factor which will interact with the promoter region. And the TF2D in turn consists of, we will move to that discussion later. TF2D consists of two factors called the TBP protein and TAF proteins or TBP associated factor proteins. So actually the subunits of TF2D which interact with this INR element are TAF1 and TAF2. So they interact with this for a successful initiation of transcription. So that's about INR element. Then the second one we are discussing is the Tatabox itself. And this is the first co-promoter identified in a eukaryotic system. And the consensus sequence is TATAA. That's hence the name what the Tatabox indicates. The other features like, as already noted, this is the site where the TF2D actually interact with the promoter region. And of the TF2D complex, the TBP protein, that means the Tata binding protein, interact with the Tatabox. That's a specific sequence, unique DNA binding sequence within the TF2D complex. So the TBP binding to the TF2 binding to the Tatabox is the demarcating step in any transcription event normally when there is a Tatabox. But you can also note that this is not a universal kind of promoter sequence. You can see only 32% of the co-promoters have been reported with Tatabox. So there are possibilities for even still unidentified sequences and factors in transcription initiation. And the importance is that why it is so dominant, because it can function even in the absence of other such sequences like BRE or INR sequences. The Tatabox alone can initiate transcription. Now the third one, the BRE. It is why it's called BRE, because this is the site where the B recognition element, that means TF2B transcription factor, will interact with the DNA sequence. So usually located at -35. The Tatabox was actually located at -25 or from -25 to -31, 32 region. So this one is located just upstream to the Tatabox, that means at around 35 base pairs upstream to the +1 site, and this is rich in GC regions. And the importance is that this is the only site of the chromosome which is recognized by a factor other than TF2D. That means this is the site recognized by the TF2B element. And when we analyzing in detail, you can see there are two sets of or two parts of this BRE sequence. One is in the upstream of the Tatabox and the other one is in the downstream, and both are important in the interaction of the TF2B to the chromosome. And now to the downstream sequences. First one is the downstream promoter element. This is usually a 7 nucleotide conserved sequence, usually located 30 base pairs downstream to the start site, and it can function even in the absence of a Tatabox. Usually the TAF9 and TAF6 will interact with this region. And finally we have the MTE. This is one of the recently discovered sequence, locates around 20 base pairs downstream, and they usually promote the interaction of TF2D to this to the promoter, or in fact to the INR element. And most of these elements can function alone, but when present together along with the Tatabox, there will be a kind of synergistic impact on the transcription process. So that's all for the co-promoter region to be discussed. Now we will be moving into the regulatory sequences, which are also important. As already noted in the beginning, these are the much important sequences for the regulation of transcription in eukaryotes. Eukaryotic transcription efficiency is not only under the regulation of the promoter sequences, which already we discussed, but also the additional sequences, what we call the regulatory sequences. And there are two kinds of regulatory sequences. That means one set of regulatory sequence which will promote transcription. That means they will be having a positive impact on transcription, and the other set of regulatory sequences are having a negative impact. They will repress the transcription mechanism. So we have certain examples of these two sets, that is the promoter proximal element and enhancers under the positive sequences, and silencers and insulators as the negative sequences. So first we will see certain features of the proximal promoter element, usually located at around 70 to 200 base pairs upstream. Then usually they help, usually what they do is they help to regulate binding of the transcription factor, that means the general transcription factors to the co-promoter region. You can see two such examples, two such sequences like GC box and cat box, which is given the name with corresponding to the sequences. So the cat box is a binding site for a cat binding protein, or CBP, or C-bus EBP, or cat box enhancer binding protein. So these are certain proteins or certain transcription promoting components or proteins. They will promote the transcription efficiency, and the GC box is also a binding site for a transcription factor called SP1. So in short, the binding of proteins at these particular sequences increases the frequency of initiation in transcription, and moreover they act as tethering elements that recruit long-range regulatory elements, such as enhancers to the core promoter region. That means enhancer sequences are important. They are categorized under the long-range regulatory sequences. So the long-range regulatory sequence will not have a direct interaction with the core region, but instead they will interact through some mediator molecules. So that mechanism is being enhanced by these proximal promoter elements. Now the second set is enhancer elements. These are still pretty away from the plus 1 side. That means around 700 to 1000 base pairs or away from the start site of transcription. You can see the noted regions. So the binding of proteins like enhancer proteins or promoter protein to these sequences are important for the initiation. And of course, for a gene, there will be many specialized transcription factors like these enhancers. That means these enhancer proteins, and these enhancer proteins can interact with these sequences in order to start the initiation. You can see the image, the transcription factors which already would have interacted with the core region and the RNA polymerase enzyme, and there are activator proteins or enhancer proteins which interact with the enhancer sequences. And you can see a corresponding example also, and this example is for the beta interferon enhancer or the beta interferon gene. You can see a many number of proteins which are coming in the interaction with the enhancer for the interaction between the enhancer and the core region. So these are the other features, like these could be located at anywhere, like in the downstream, upstream, or even within an intron, and can function without or irrelevant of the orientation of the sequence is irrelevant, and having a length of around 500 base pairs. But the other last point is important, that these will be either tissue specific or developmental stage specific. That means all such sequences are present in all set of genes. It's not like that, but certain set of or certain sequences are present in certain tissues and also maybe in certain stage of growth. So in other cells that will be completely absent. Now to silencers and insulators. These are the representative examples for the sequences which are repressing transcription. And silencers are acting almost similar to that of enhancer sequences. They are located at various locations, various sites in the chromosome around the gene location, and they will have a negative impact. So which means that when a silencer is present, they'll block gene expression or they'll block the transcription initiation. And insulator is also important because the insulators will block transcription initiation in such a way that they'll block the chromosomes to unwound. That means the opening of the organized chromosome will be prevented when there is an insulator sequence present. And typically they are having a length of around 300 base pair to 2 kb in length. And there are two functions, or there are two kinds of insulators available. One is called the chromatin boundary marker. The other one is the enhancer blocking activity. Right. So the first one is that means an insulator marks the border between regions of heterochromatin and euchromatin. You know that. So euchromatin is the part where you have the genes, whereas heterochromatin is normally in a condensed state. So these two parts has to be clearly demarcated, and this is being done by the insulator sequences, which will prevent the high condensation of heterochromatin which is spreading into the euchromatin. So that will be prevented by the insulator. Similarly, they also prevent an inappropriate cross activation or repression of a neighboring gene by blocking the action of enhancers and silencers. Right. So that means the enhancer activity of a particular gene will be limited to that gene alone. The activity spreading to a second gene or a neighboring gene will be prevented by the presence of an insulator. Yes, so that is the end of that part. Now we are moving into the discussion to the most important element, the RNA polymerase enzyme. So in bacteria we have seen certain features of RNA polymerase of the bacterial system, but in eukaryotes, as already we have seen that there are three different types of RNA polymerases specifically for the different types of RNA molecule, like type 1, type 2 and type 3 for the rRNA, mRNA and tRNAs respectively. So these RNA polymerase enzyme was initially introduced or discovered by the team of scientists led by Roger Kornberg in 1980s, and that enzyme particularly they studied was the RNA polymerase 2, and which is the most studied and well characterized enzyme, and hence we'll confine our discussion to the RNA polymerase 2, right? And when we see the structure, you can see that image. There are 12 different subunits. They are denoted like RPB1 to RPB12. So like in bacterial RNA polymerase enzyme, we have the core region and also some additional sequences. The core region consists of 10 different subunits, and the core region plus an heterodimer usually comprise the holoenzyme. And this heterodimer is there are subunits for RPB4 and RPB7, which is not part of the core enzyme. And this is the comparison between comparison of RNA polymerase subunits between the eukaryotic, archaea and E. coli system. You can see the pore 2 consists of 12 different subunits. So all those 10 subunits comprising the core and the RPB4 and 7 as the additional sequences. Again, you can see the comparative representation, and these are the homologous. The color indicates the homologous part. And the core enzyme require the additional heterodimer. That means the 4/7 complex is required for the catalytic activity of the enzyme. And when we see the different features, one of the most important feature to be discussed in terms of their function is a carboxy terminal domain. That means of the different subunits in the RNA polymerase, we have the largest subunit as noted as the RPB1, just like the beta dash subunits in bacterial polymerase. The RPB1 is the largest subunit, and the carboxy terminal domain of the RPB1 is essential and important for the RNA polymerase activity, or for clearly to the transcription mechanism. So which contains, that means the CTD of the RPB1 contains a stretch of 7 amino acids which are conserved, and usually these are having a repeated sequence of what that given the sequences like tyrosine, serine, proline, threonine, serine, proline and serine. This is the heptapeptide sequence, and these sequences are repeated multiple times. Usually in humans you can see around 52 times of these heptapeptide sequences at the carboxy terminal domain of the RPB1 element, which is the largest subunit of the RNA polymerase 2. And what is the importance of this sequence? It is because for the initiation to occur, that means for the successful binding of the different subunits of the RNA polymerase enzyme to the promoter region, we need an unphosphorylated carboxy terminal domain. That means in the beginning, all the CTD, all the sequences at the carboxy terminal domain must be unphosphorylated. Whereas for initiation, after initiation, for elongation process, you need to phosphorylate these CTD sequences. So the CTD domain is important because the phosphorylation and dephosphorylation controls initiation and elongation events, right? So out of the heptapeptide or that 7 amino acids, the serine residues are being phosphorylated, usually at position 2 and 5, and then occasionally at 7 also. So there is a phosphorylation of serine residues usually by kinase enzyme, and precisely when we discussed the transcription, the TF2H is having a kinase activity, and the TF2H will do the phosphorylation event along with other kinases, and the dephosphorylation for the recycling of the factors will be done by another phosphatase enzyme called FCP1, so that these molecules could be used for the next cycle. Now we are moving into the third and to the next component called the general transcription factors. In a eukaryotic system, the RNA polymer the enzyme require assistance of a set of usually 5 or 6 different general transcription factors, which are responsible for the one is the promoter recognition, and second one is unwinding or the opening of the duplex DNA at the promoter site. So the different such factors are given. They are collectively called as the general transcription factors. You can see them as the TF2A, TF2B, TF2D, E, F and H are the important elements, of which TF2A is of least important. All other sequences are required for a successful mechanism in vitro. So the number of subunits are also given there. TF2A in 2, 2B in 1 number, TF2D with internally the TF2D consists of 2 components. One is the TBP called Tata binding protein, which will be present in 1 unit, and there are around 14 different additional proteins called TAFs or the TBP associated factors. So there are 13 to 14 different subunits as TAF 1 to 14, and there is 1 TBP in the TF2D, right? And there are 2 TF2E and 2 TF2F, 3 F complex and around 10 H subunit. So the H is also important, and we will see that later. See, first we will move into the TF2D. So TF2D is the first general transcription factor to interact with the template DNA, and followed by the rest of the transcription factors for initiation. So as for any biochemical reaction, the first reaction is quite important, and here also this interaction is important, and this is one of the rate limiting step in transcription. So once the TF2D begins to interact with the promoter region, then the system will commit for transcription. And already stated, this consists of TBP protein and TAF protein, together what we call the TF2D. And the TBP is the sequence specifically for interacting with the Tatabox in the promoter region. And also not only to the Tatabox, as already stated, they can also interact with some of the neighboring sequences also. And this can also mediate the transcription of a gene even there is no Tatabox. That will be a question, because when the TBP is there and which interact with the Tatabox, and suppose already we have stated that there is only 32% of genes with the Tatabox. So suppose if a gene do not have a Tatabox, then what will be the consequence, or how the transcription initiation occur? And the answer is that the TAF will take the charge. The TAFs can usually interact with sequences other than the Tatabox, right? So they are supporting the interaction of TF2D to the promoter regions without a Tatabox. Fine. So the interaction will occur even if there is no Tatabox, right? So the TBP Tatabox complex formation and the nucleosome formation are mutually exclusive event. This is an important point we have to note. That means we know that in a eukaryotic system, the chromosome is in an organized manner. There will be chromatid formation. There will be different levels of organizations are there for the eukaryotic chromosome. So and all those organization levels starts with what? A nucleosome. A nucleosome is the basal unit of the chromatin organization. So what the statement says, TBP Tatabox complex formation and the nucleosome formation are mutually exclusive. That means the TBP TF2D complex cannot bind to the DNA which is part of a nucleosome. That means we know that there are nucleosomes usually spanning a distance of around 200 base pairs. So within that region of a chromosome, the TF2D cannot interact. That's important. And second, or otherwise also you can say vice versa. That means when there is a TBP Tatabox complex occupied already in a chromosome, the histones cannot occupy that place. So these are two mutually exclusive events. That means the histone interaction with the chromosome and also the TF2D interaction with the chromosome. So it is clear that the chromatin has to be decondensed well before the TF2D binding for a normal initiation. Otherwise, it cannot initiate transcription. So again, when we move in detail, we can see a lot of things. I'm just taking 1 or 2 points more. There are many factors controlling the recruitment and release of TF2D. Right, the interaction and release are important. Two such examples are NC2 and MOT1 factors. NC2 is nothing but negative cofactor 2 and MOT1 is modifier of transcription 1. These are models taken from yeast organism. NC2 can bind to the complex, that means TBP Tatabox complex, so that the premature association of TF2A and B will be prevented. Right, that means which provide enough time for the interaction of the TF2D to the core promoter region before the TF2A and B is coming in place, and also which helps the TBP to dissociate from the Tatabox and to slide along the DNA. That means it helps in elongation process. And similarly, MOT1 also helps in the dissociation of TBP from the DNA molecule. Fine. So now move to the other factors, TF2B and TF2A, of which TF2A is of least important as it is not required for when it is reconstituted in vitro. So we will not be discussing much about TF2A. But usually B and A elements are recruited almost simultaneously after the TF2D interaction recruitment to the promoter site. So the TF2B, the significance is that which orient the complex on the promoter and set the direction of transcription. So in which direction the transcription has to occur is being set by the TF2B protein. Usually they have multiple binding sites. At one end they bind to the TBP and in the other end they bind to the GC reach already noted BRE element. Right. And also provide an information on which of the two template DNA which has to be used as the template precisely. And also moreover help in the conversion of the closed promoter to an open promoter. Then TF2F. Usually this factor is usually coming along with the RNA polymerase enzyme, because the RNA polymerase alone cannot be stabilized or they cannot form a stable complex in the promoter region. So the TF2F is important as it is required for the polymerase to be recruited to the promoter region. And binding of this RNA pole to TF2F complex stabilizes this entire ternary complex. That means now there will be the DNA Tata binding protein, TF2B complex, the TAFs will be there. Now the incoming RNA pole 2 and TF2F complex. So now this complex is what we call the pre-initiation complex, and to this pre-initiation complex there will be recruitment for TF2E and TF2H, which are actually the last stage of initiation process or the beginning of elongation process. So the TF2F is a heterodimer. When we analyze the structure, it's a heterotetramer which is composed of TF2F alpha and beta in 2 units. right? Then the TF2E is a heterodimer again. It has a role in the recruitment of TF2H. That is the importance. Since TF2H is important, the recruitment of TF2H becomes so important for the transcription process. And together with TF2H, TF2E also is required for the ATP dependent formation of an open complex prior to the formation of the, or prior to the first base to be added, or the phosphodiester bond formation between the bases, right? And finally the TF2H, this is one of the very important, very significant molecule which is having 10 different subunits usually recruited into the site, and having a heavy molecular weight of around 500 kilodalton. It's just almost equal to that of an RNA polymerase enzyme. And this is the only transcription factor with an enzymatic activity. So this TF2H consists of enzymatic activity or enzymatic domains. And there are two such domains. One is the helicase activity domain. The other one is a kinase activity domain. Fine. So the helicase activity, of course, as you know, it's for the unwinding of the helic, and kinase activity for the phosphorylation, dephosphorylation mechanism. So this is what we noted as the pre-initiation complex. There will be all those different subunits along different components of the transcription factors along with the RNA polymerase enzyme. So the order of interaction, in short, you can say the first one will be D followed by A, B, then the polymerase enzyme along with F complex, then E and H. So in short, you can say D, A, B, Pol, F, E, H. So that will be the order for the pre-initiation complex formation or the initiation complex formation. Right. So now moving into the last part, the transcriptional coactivators and co-repressors. So these are the proteins usually interacting with the enhancer sequences and other such long acting regulatory sequences. So as already we have stated that in eukaryotes, the chromatin is in a highly condensed pattern. So the decondensation has to be done according to the communication, according to the signal received by the system. So that decondensation is mediated by the proteins like the coactivators and co-repressors. So these are proteins that increase or even that can decrease transcriptional activity through protein-protein interaction. That's important. They are not associated with the DNA directly. So we have two such examples. One is the chromatin modification enzymes or the chromatin modification complexes, of which we can see many examples like histone acetyl transferases, histone methyl transferases, and the process called ubiquitinylation, then phosphorylation and ribosylation, sumoylation. So all those different reactions are reactions involved in the chromatin modification. That means usually they are involved in the condensation decondensation process. So I am not going into the discussion of that part, that we will discuss later in some other video. And the second one is chromatin remodeling complexes. So all these proteins or all these different coactivators and co-repressors are doing a crucial role in decondensation mechanism. And again, another set of proteins called mediators, they act as a molecular bridge. So these are additional factors that connect transcriptional activators which are bound at enhancer sequences or other such regulatory elements with the RNA polymerase enzyme. And moreover, they can stimulate the kinase activity of TF2H, and these are unique to eukaryotic system. You cannot see any such sequences in a bacterial transcription machinery. And this image will show you how the decondensation occurs. So this will be the initial status of a chromosome. If there is no gene expression, you can not like the gene is in off stage, right? It is the condensed form of chromatin. So when you want to own the gene, or when you want to undergo a transcription within these particular region, so you have to use, or there is a role of various additional proteins called the activators, coactivators, mediators, repressor, co-activators or co-mediators. So all these molecules together will do the decondensation. So the decondensation is important, and the decondensed chromatin can only undergo a transcription process. So this is how the transcription begins in a eukaryotic system. So this will give you an idea about the transcription machinery which are common in most of the eukaryotic transcription system. So now we are going to wind up this part of the presentation. So we have discussed about the core promoter regions or the core promoter sequences, the regulatory sequences, that means the additional sequences other than the core promoter sequences in eukaryotic chromosome, and about the RNA polymerase enzyme, their structure and functional features. Then finally the additional proteins like coactivators, co-repressors and mediators, which are supporting or which are regulating the transcription process in a eukaryotic system. So, and I hope you would have understood the various mechanisms and various proteins and sequences involved. And once again, thank you for watching.